Heredity and Evolution. Introduction to Heredity and Evolution. The living world is composed of two major groups of organisms, namely the prokaryotes and the eukaryotes. Prokaryotes are a group of organisms that lack a nucleus and other membrane-bound cell organelles. Eukaryotes, on the other hand, comprise of cells with a very well-defined nucleus and even show the presence of complex structures enclosed within membranes. The main difference between prokaryotes and eukaryotes is that, in the case of prokaryotes, the nuclear material is not enclosed by a nuclear membrane but is in direct contact with the cytoplasm. In eukaryotes, the nuclear material or the nucleus is enclosed by a double membrane called the nuclear membrane. The nucleus consists of genetic material which is organized in the form of very fine, thin thread-like fibers called the chromatin fibers. During cell division, the chromatin fibers condense and become thick and ribbon-like and are known as chromosomes. Each chromosome, in its condensed form, consists of two chromatids joined at some point along the length. Chromosomes are formed of DNA, that is, deoxyribonucleic acid. The genetic information is stored in long stretches of DNA in the form of units called as genes. A gene is thus a specific length of a DNA chain which holds the information to control a specific characteristic of an organism. For example, features such as color of hair, rolling or folding of tongue in human beings, etc. There are thousands of genes on a chromosome which control various characteristics of an organism. Each gene is assigned a specific location on the chromosome which eventually determines the function of that gene. Now let us understand the mechanism of gene function with the help of an activity. Have you ever observed rose plants growing together in a garden? Do they appear similar or do they look very different? Well, when we observe the rose plants in a garden, all of them appear similar. This is because each of these plants is propagated through cuttings from the same parent plant. Similarly, all sugarcane plants growing together in a sugarcane field will also be similar. Plant cutting, also known as striking, is a mode of vegetatively propagating plants through the method of asexual reproduction. The rose plant and the sugarcane plant both can be propagated through stem cuttings. In the case of rose, we take cuttings from the tip of the stem of a mature mother plant, while in the case of sugarcane, we take cuttings of comparatively smaller pieces of stem. The rose cuttings are planted vertically, while the sugarcane cuttings are planted horizontally. Due to the similarity in their method of propagation, these plants do not show much variation in their growth pattern. Now, let us study one more example. Each individual is different from the other with respect to characters such as height, weight, body stature, color of hair, etc. They differ even in terms of their daily habits. From these examples, we understand that asexual reproduction results in small amount of variations as compared to sexual reproduction. Why do these variations occur? Let us understand this further. Let's do a quick check. accumulation of variation during reproduction. In case of humans, the sexes are separate. Human beings reproduce through sexual reproduction. A haploid male gamete fuses with a haploid female gamete to form a diploid zygote with 2 sets of chromosomes. The zygote formed after fertilization receives 1 set of chromosomes from the mother and the other set from the father. Thus, there is a recombination of genetic material which ultimately results in variation. A child has certain features similar to that of the mother and certain features similar to that of the father. The offspring thus closely resemble their parents. Do you know how variations present in the parents are passed on to their children? Let us explore this further. The process of reproduction gives rise to new individuals. The offspring so produced are similar to their parents in many essential characters. However, they do show some minor differences. These differences in the characters of the individuals of a species are called variations. The variations produced in the organisms get accumulated. They are passed on from the parents to their offspring, that is, from one generation to the other. This phenomenon is known as inheritance, and the variations are called inherited variations. The members of the successive generation inherit the characters of the previous generation. The significance of a variation shows up only if it continues to be inherited by the offspring of several generations. The inheritance thus essentially provides the general basic pattern of the body with some minor differences for the next generation. When the individuals of the new generation reproduce, their offspring will have the differences that are inherited from the previous generation as well as certain newly created differences. These differences are the variations produced by the reproductive process. Let us understand this concept with the help of an example. Look at the figure carefully. What do you observe? The figure illustrates an example of a single organism undergoing reproduction as seen in asexual mode of reproduction. The parent organism reproduces and gives rise to 2 new individuals. These 2 individuals, although similar in body structure with the parent organism, do have some minor differences. Further, in the next generation, the new offspring reproduce individually to produce 2 new organisms each. These 2 organisms also resemble their respective parents, but do have certain minor differences. Some of these differences are unique, while some of them are inherited from their parents. Thus, when we observe all 7 organisms together, starting from the original parent, the 2 offspring and their offspring, we notice that they are all similar in body structure, but do show certain minor differences. Do you know why in asexual reproduction the individuals have only a few differences between them as compared to sexual reproduction? Well, in asexual reproduction, the individuals produced are quite similar to each other and exhibit minor differences among themselves. This is because of small inaccuracies involved during the process of DNA copying. In this way, diversity is created in the subsequent generations. This is observed in all organisms which reproduce through asexual methods. For example, bacteria and eukaryotic algae. In contrast to these, sexual reproduction involves fusion of 2 different germ cells. Thus, greater variations and hence greater diversities will be generated. Let's do a quick check. Heredity. Inherited traits. Passing on or transmission of physical or mental traits from the parents to the offspring is known as heredity or inheritance. Why does a rat give birth to a rat only and not a cat? This is because the organisms reproduce their own kind and transfer their characters from one generation to the other. That is why a rat will produce young rats similar to itself. The new offspring will exhibit the same body design with a few variations due to some minor differences in them. Living organisms follow the universal law of like begets like. It means that the offspring of dogs will be dogs, pigeons will produce pigeons, and humans will beget humans only. We have learned that organisms reproduce and produce their own kind, but the offspring produced do not look exactly like parents, and thus it can be said that human population shows a great variation. Let us perform an activity to understand this. Activity. Observe the ears of all the students of your class. Prepare a list of students having free earlobes and those having attached earlobes. Calculate the percentage of students falling in each category. What is your observation? Well, the percentage of students with free earlobes and those with attached earlobes is 40% and 60% respectively. Now, if you were to observe the earlobes of the parents of these students, you will observe that their pattern of earlobes is the same as that of their children. This character is thus inherited from the parents. Such characteristic features which are inherited from one generation to the other are called traits. These traits are passed on from one generation to the other. Let us now proceed to understand the rules for the inheritance of these traits. Mendel's rules of inheritance. Inheritance is the transfer of hereditary characters or traits from parents to the offspring. The offspring resemble their parents but show certain minor differences. These similarities and differences are because of heredity. The credit for the concept of heredity goes to Sir Gregor Johann Mendel, who was the first person to carry out work on inheritance. Mendel was not only a priest in a monastery but also a naturalist. He conducted several experiments in the monastery garden to study transmission of certain traits in pea plants. He is now recognized as the father of genetics due to his remarkable contribution to the field. Several other scientists had studied the inheritance of traits. However, Mendel blended his knowledge of science and mathematics and was the first person to give the hypothesis of the existence of a factor called the Mendelian factor to explain continuity and variation. This factor is known as gene. He conducted experiments on pea plants and studied the inheritance of certain traits. He concluded that the inheritance of these traits follows a particular pattern. The significance of Mendel's work was not recognized until the turn of the 20th century. In 1900, scientists rediscovered Mendel's work and its importance in the field of genetics. His work was thus appreciated by the society after his death. Based on his work, Mendel put forth certain laws which later became famous as the laws of inheritance. The laws formulated by Mendel constitute the foundation of the modern science of genetics. Let us now begin our study of Mendel's laws with the help of experiments involving the breeding of native pea plants. Mendel's law for inheritance of traits. The laws of inheritance of traits in human beings are related to the fact that both the father and the mother contribute practically equal amount of genetic material to the child. This implies that each trait can be influenced by paternal as well as maternal DNA. If the contribution of both the father and the mother is equal, then for each trait there will be two alternate forms for each character, producing different effects in the child. These alternate forms of a gene are called alleles. Mendel thought on similar lines and conducted experiments on the pea plant that reproduces through sexual reproduction. Based on his observations and results, Mendel formulated the laws of inheritance. Mendel performed his experiments over a century ago, but the results and findings are still very much relevant to modern genetics. It is very interesting to study these experiments. Do you know which plant was selected by Mendel for his work? Mendel selected plants of Pisum satirum, commonly known as garden pea, for his experiments. Mendel chose pea plants because they exhibited clear-cut differences in certain characters which were easy to tell apart. The pea plant has several distinct varieties, each with visible contrasting characters that appear on separate chromosomes. For example, height of plant, coloration of flower, coloration of seed, etc. Let us study Mendel's experiments involving a single pair of contrasting characters in detail. During the course of his experiments, Mendel conducted many crosses involving plants with a single pair of visible contrasting characters. For example, tall and dwarf plants, red and white colored flowers, round and wrinkled seeds, green and yellow colored seeds, etc. are some of the characters. Such a cross, which involves only a single pair of contrasting characters, is called a monohybrid cross. Let us understand this cross with the help of an example. Mendel selected one pea plant bearing red flowers and another pea plant bearing white flowers as parents to perform a monohybrid cross. These plants are considered to be of parental generation or P1 generation. The terms like dominant and recessive were used by Mendel for red and white flowers respectively. Do you know what the words dominant and recessive imply in genetics? A dominant character is that character which hides or masks the expression of its corresponding character, which then becomes recessive. From a single pair of contrasting characters, one will be dominant and the other will be recessive. The red flowering varieties of pea plants were called dominant because cross-pollination of red flowering plants with white flowering plants resulted in offspring with red flowers in the successive generation. The offspring did not exhibit a single white colored flower. The dominant red color did not allow the expression of the recessive white color. Hence, white flowering plants were called recessive. These findings helped Mendel to formulate one important law of inheritance called the law of dominance. It states that in every pair of contrasting characters or alleles, one is more frequently expressed than the other. This allele is referred to as the dominant allele, and the other, which is less likely to be expressed, is known as recessive allele. Likewise, Mendel studied 7 pairs of contrasting characters. Let us discuss about these characteristics. When we consider the shape of a seed, round shape is a dominant trait, while wrinkled shape is a recessive trait. respect to seed color, yellow color of seed is dominant and green color is recessive. When we consider the color of the flowers, violet or red color is a dominant trait and white color is a recessive trait. With respect to fruit or pod shape, inflated shape of pod is a dominant trait, while constricted shape of pod is a recessive trait. In terms of pod color, green color is dominant and the yellow color of pod is recessive. With respect to position of the flower, axial position is a dominant trait, while terminal position of a flower is a recessive trait. When we consider the height of stem, tallness is a dominant trait, while dwarfness is a recessive trait. Mendel's experiments involved only pea plants with dominant and recessive traits. Through his experiments on pea plants, Mendel deduced that the factors or genes responsible for a particular character were present in pairs, one as dominant and the other as recessive. A particular convention exists for representing the dominant and recessive factors. Dominant genes are represented by capital letters, while the corresponding recessive genes are represented by corresponding small letters of the alphabet. For example, capital R is for red flower and small r is for white flower. Genotype is the genetic constitution of an organism. It is the description of genes present in an organism. Phenotype, on the other hand, refers to the observable characteristic of an organism. For example, red flowering plants. Now, since the genes occur in pairs, the genotype of red flowering plants will be capital R, and that of white flowering plants will be small r. In sexual reproduction, these factors or alleles segregate during the process of gamete formation. This results in the formation of two types of gametes, one with capital R factor and the other with small r factor. Based on this observation, Mendel formulated his first law of inheritance called the law of segregation, which states that the alleles that make up a gene separate from each other or segregate during the formation of gametes. The generation of offspring produced by P1 generation of parents is called as the first filial generation or F1 generation, and the new form of offspring resulting from a cross or breeding are called as hybrids. Thus, phenotypically, when a white-flowered parent P1 plant is cross-pollinated with a red-flowered parent plant, the first filial generation F1 generation produces plants with red-colored flowers. Red coloration is dominant and masks the effect of white coloration. That is, when parents having genotype small r are crossed with parents having genotype capital R, two types of gametes are produced, namely capital R and small r. By the combination of gametes, we get F1 generation of plants with genotype capital R, small r. The first filial generation, F1. During his experiments with pea plants, Mendel noticed that all the plants of F1 generation had flowers that were red in color. These red flowering varieties of plants were different from those of P1 generation. From these observations, Mendel concluded that red color was dominant over white. Although the F1 individuals were bearing red-colored flowers, they also contained factors responsible for the development of white color. That is, their genotype was capital R, small r. These F1 plants produced red flowers which were clearly visible. That is, the external appearance or the phenotype was red. Thus, these F1 plants are phenotypically red but genotypically hybrids with a combination of capital R, small r. In this experimental cross, red-colored F1 plants are similar in their phenotype but different in their genotype in comparison to the red-colored parental P1 plant. That is, P1 plants are capital R, and F1 plants are capital R, small r. Mendel was curious to find out the result if he crossed two F1 individuals having this genetically hybrid combination. Mendel thus continued his experiments with these F1 plants. These F1 hybrid plants during gamete formation produced two different gametes, namely capital R and small r. He crossed the plants among themselves or carried out self-pollination, that is, capital R small r cross capital R small r. This resulted in a successively new generation different from the previous one, which is called as the second filial generation or F2 generation. The second filial generation, F2. The F2 generation resulted from selfing of F1 plants, showed the presence of both red-colored flowers as well as white-colored flowers. Mendel experimentally observed that out of a total of 929 progeny, about 705 plants bore red flowers while 224 bore white flowers. If we calculate the ratio of the same, it is approximately 3 parts red is to 1 part white, or simply 3 is to 1. When we calculate the genotypic ratio, it is 1 capital R is to 2 capital R small r is to 1 small r. Thus, phenotypically, there are two types of progeny, namely red-flowered plants and white-flowered plants, while genotypically, there are three types of progeny, namely F2 pure dominance with constitution capital R and producing red flowers, F2 pure recessive with constitution small r and producing white flowers, F2 hybrids with constitution capital R small r and producing red flowers due to the dominant gene capital R. The F2 pure dominance and pure recessives are both homozygous, showing the presence of two same types of alleles in the zygote after fertilization. The F2 hybrids are, however, heterozygous, showing the presence of two different types of alleles in the zygote after fertilization.